Good afternoon, everyone, and welcome to the NERIC Lightning Talk sessions. My name is Samantha Meenick, and I'm from the University of Rhode Island, and I'll be moderating the technology session today. Prior to beginning, I'd like to let everyone know that the recording for the National Association of IDEA Principal Investigators, or NAPI, update by Dr. Gus Kuselos is on the website since there were technical difficulties yesterday that prevented him giving that presentation. With that being said, we're excited to have 14 talks for the technology session this afternoon, in addition to the other talks being given in the other sessions and the ones following. Each presenter will have 3 minutes to discuss their research before we move on to the next talk. So this will be very fast, rapid talks. We invite anyone listening in to type questions into the Ask Your Question area on the right of your screen, making sure that you list which presenter the question is for. And we will hopefully have time at the end of each hour-long session for a few questions, which I will ask randomly. So our first talk this afternoon will be given by Arvind Sopriyatna from Brown University. Arvind, please begin when you are ready.
Good afternoon, everyone. My name is Arvind Sopriyatna, and I am a postdoctoral fellow in the Coulomb Lab for Cardiovascular Regenerative Engineering at Brown University. Today, I will be talking about how our lab utilizes human 3D cardiac microtissues that enable chamber-specific arrhythmic risk assessment to improve the safety standard of drug screening. Cardiotoxicity remains to be one of the leading causes of not only drug withdrawal from the market, as shown on the pie chart on the left here, but also high attrition rate during drug development. We know that many drugs work by targeting specific ion channels, and that although both the atria and the ventricles share many of the same ion channels, with the exception of the 2 channels outlined in green here, their expression levels are quite different. This means that the same drug could elicit a different chamber response at different dosages. However, a problem with current drug screening platform is that they primarily focus on ventricular responses to drugs, while atrial responses continue to be overlooked, despite atrial arrhythmias being the most common type of arrhythmias in the clinic. And so our goal here is to develop a sensitive in vitro platform for chamber-specific evaluation of arrhythmic risk. Next slide, please. Our approach begins with differentiating cardiomyocytes from human-induced pluripotent stem cells by modulating the Wnt signaling pathway. We supplemented retinoic acid to generate atrial cardiomyocytes and employed lactate purification to increase our cardiomyocyte purity. To generate 3D cardiac microtissues, we basically pipetted a mixture of cardiomyocyte with 5% human cardiac fibroblasts into agarose hydromoles with 35 micromoles. We then electrically stimulate them under 1 Hz pacing for 1 week as the microtissues undergo significant compaction. Optical mapping was then used to characterize action potential differences. As seen by the traces here, ventricular microtissues showed the characteristic spike and dome shape, whereas atrial microtissues have a more triangulated waveform. Notably, we detected slower rise time, shorter APDs, and greater triangulation in atrial microtissues. We also showed that our atrial microtissues are spontaneously active, whereas our ventricular microtissues are quiescent. Immunohistochemistry also confirmed successful subtype specification by double staining for atrial marker MLC2A shown in red and ventricular marker MLC2V shown in green. Next slide, please. Finally, we tested our microtissues with several drugs that target different ion channels. The first drug is 4-aminopyridine, which is a candidate drug for atrial fibrillation, which blocks the atrial-specific ultra-rapid potassium channel IKUR at low doses. We were able to detect dose-dependent prolongation in APD and our atrial microtissues, and this response was absent in our ventricular microtissues. We also tested ibuprofen, which is a candidate drug for AFib suppression that blocks the funny channel. And likewise, we detected a dose-dependent increase in spontaneous activity cycle length in atrial microtissues. The final drug, E4031, is a hERG channel blocker, and although not a drug for treating AFib, we wanted to highlight that we can also detect arrhythmic events like early after depolarization in our platform, as shown by the red star in the traces. In conclusion,
to wrap up.
Okay, in conclusion, our human 3D cardiac microtissues platform replicates dose-dependent atrial and ventricular drug responses, allowing for robust cardiotoxicity screening and novel drug development. And with that, I want to thank everyone in the Coulomb Lab, as well as our funding sources from NIH, NSF, and AHA. Thank you.
Thank you. Very nice presentation, Arvin. I tell you, it goes really fast. So next up, we have Ishnur Sidhu from University of Delaware. Ishnu, begin when you are ready.
Hello, everybody. I'm here to present to you a 3D hydrogel system for modeling hematologic malignancy known as transient myeloproliferative disorder using iPSC technology. TMD is a unique pre-leukemic stage affecting 10% of the Down syndrome neonates with 20% mortality rate due to multi-organ failure. 30% of these kids develop acute myeloid leukemia in their first 5 years of life. TMD disease progression is a multi-step where the first hit is the presence of extra copy of chromosome 21. The next step is the mutation in the X-linked gene GATA1, producing a truncated and oncogenic form of protein called as GATA1S. Since this disease affects very young kids, it is important to understand the disease to develop novel therapies. Next slide, please. The first goal of the study was to develop a culture system to provide cell-to-cell and cell-to-matrix interaction during hematopoietic differentiation of iPSC, which is not possible to study in 2D culture system. The 3D hydrogel in the study used a PEG backbone decorated with an integrin binding and an enzymatically degradable peptide. The PEG hydrogel supported the viability and pluripotency as shown in the figure on the left, with the 3D colonies being more spheroidal in shape than 2D cultures determined by the aspect ratio calculations. Subjecting the hematopoietic iPSCs to hematopoietic differentiation, round HSPCs or the hematopoietic stem and progenitor cells were collected on day 12, as shown in the figure on right. The immunophenotype analysis using flow cytometry revealed 1.7-fold higher percentage of early HSPC and multipotent progenitor cells generated from 3D compared to 2D. This was further corroborated by the presence of higher number of mixed colonies generated from the 3D HSPCs, as shown in the figure graph on the right. Next slide, please. Next, the PEG hydrogels were utilized to model TMD using isogenic, disomic, and trisomic 21 iPSCs. The CRISPR-Cas9 technology was used to generate mutation in the GATA1 in trisomic background. The ploidy analysis was done to bioauthenticate the iPSC lines. The hematopoietic differentiation of the 3 lines revealed that while the trisomy alone augments the erythroid and megakaryoid population compared to the disomic counterpart shown in blue, the mutation in the GATA1 drastically reduced the erythroid population, meanwhile enhancing the myeloid and megakaryoid population compared to the wild-type trisomic HSPCs. The immunoblot analysis revealed the exclusive presence of GATA1S in the mutant cells. The GATA1 mutation leads to generation of only myeloid-rich colonies in the colony-forming assay, while the trisomy alone has an increased number of erythroid and mixed colonies. In summary, we demonstrated the potential of PEG hydrogel as a platform for 3D disease modeling, featuring the salient features of transient myeloproliferative disorder. With that, I would like to thank INBRE to give me an opportunity to present my work and all my funding sources. Thank you.
Thank you very much. So if I'm not mistaken, we have 2 speakers who are currently missing. And so, Noelle, we're going to be going forward with you in just a second once the slides come up. So next, I'm pleased to present Noelle Kozarek from the Graduate School of Medicine at Dartmouth. And Noelle, we're ready to hear what you have to share.
Good afternoon, my name is Noelle Kozarek, and today I'll be presenting my research on the identification of distinct fibroblast populations in a systemic sclerosis 3D skin model using single-cell omics. To begin with, systemic sclerosis, or SSC, is a rare autoimmune disease characterized by fibrosis of the skin and internal organs, vascular abnormalities, and autoantibody formation. You could see some of those vascular abnormalities in this photo on the left here of Raynaud's phenomenon. Patients will also develop sclerodactyly, so that's when their skin contracts so tightly that it causes this claw-like feature. Further, mouse models fail to recapitulate all the systemic features of this disease, and human samples are often difficult to come by given the rarity of this disease. So the goal of this study is to characterize fibroblast diversity, which are those collagen-producing cells, in a 3D model of healthy control and SSC human skin using single-cell RNA sequencing. In order to do this, we construct a self-assembled skin equivalent, or SASE tissue model. It takes about 5 weeks to make these tissues, and we add monocytes and fibroblasts, as well as plasma that are all autologous, as well as foreskin keratinocytes to this model. And on the bottom here, you can see on the left that we have some control tissues, and on the right, we have some SSC tissues. Next slide, please. Our main hypothesis was that we were going to observe a lot of fibroblast diversity in this model, and it seems that we have. We identified 4 distinct fibroblast subpopulations in this model, as well as a small population of keratinocytes and macrophages. We were able to identify each of these cell types based upon the canonical markers, such as VIM and Thi1 for fibroblasts. It's interesting to note that we observed proportional differences in the SSC samples compared to the healthy controls. And we noticed this based on frequencies as well as odds ratio tests. So in the fibroblast ApoE CFD cluster, as well as the fibroblast STC1 VEGF-A cluster, there were proportionally more cells in those clusters in the SSC samples compared to healthy controls. Next slide, please. We also did extensive differential gene expression analysis as well as GSEA on this data. And we found that TNF alpha signaling, hypoxia, and PI3 kinase signaling, which are important in fibrosis, exhibited positive proportional differences in SSC compared to healthy control. And finally, in the fibroblast STC1 VEGF-A cluster, it exhibited a high proportion of expression of inflammatory genes. In the LUM TPM1 cluster, there was a high proportion of collagen genes expressed. And finally, in MMP2 PTGDX, the matrix remodeling genes were highly upregulated in this cluster. So essentially, what we concluded from this was that these cell populations represent targetable populations for therapeutics. And with that, I would like to thank the Whitfield Lab, especially Tamar Abel, the Puli Lab, especially Hee-Tek Yang, and by funding the BD2K and Dartmouth QBS program. Thank you.
Excellent. Thanks, Noelle. That was right on 3 minutes. So next up, we have Bader Jirai from the University of Delaware. Bader, please begin when you are ready. You're muted.
Thank you. Thanks for the opportunity to present. My name is Bader Jirai, and I'm a 5th-year PhD candidate at the University of Delaware. My talk is on using nanoparticles to enhance the survival of primary macrophages. And so, as you know, macrophages are important cells to study because they have many different therapeutic functions. They can phagocytose and destroy pathogens, and they can present antigens to adaptive immune cells and promote immunity, and they can be activated into pro or anti-inflammatory states that have therapeutic implications in cancer and autoimmune diseases. And so here I'd like to focus on ex vivo macrophages that are extracted from tissue. As you can see in the figure to the left, macrophages can be isolated, activated in a certain way, and then administered back to the body, similar to CAR T-cell therapy. But here, macrophages have the distinct advantage in that they don't have major challenges when it comes to solid tumors. So that's really a major benefit. And also to the right, we have vaccine and drug screening, especially in the case of personalized medicine. But the reason we don't see macrophages in the clinic because they have very poor survivals, both ex vivo when they're taken out, but they're also administered back into the body in vivo. So next slide, please. And we have discovered that when macrophages are treated with inert polyethylene glycol-based nanoparticles, they can remain surviving in culture for significantly long periods of time. So as you can see on the top, macrophages are dying very rapidly, but when they're treated, and then you see in the microscopy images on the bottom, they can show very nice survival rates, which are quantified to the right in that figure. And so this really shows us as a method to enhance the survival and potency of macrophage-based cell therapy just by means of treating them with these nanoparticles. And obviously, we've tracked some pro or anti-inflammatory markers that you see in the bottom, and we don't see any major changes into those. These are just select markers, but we've screened a wide range of markers, and this is really the benefit of using a material like polyethylene glycol-based nanoparticles. Next slide, please. We've also showed that this effect is seen with all sorts of nanoparticle chemistries. Here we have screened a wide range of nanoparticle chemistries. We always see this effect, obviously in different ways that we can, or different ratios that we're beginning to characterize why that is. And obviously this is inherent to the nanoparticle properties. We've also screened different types of macrophages, in this case alveolar macrophages. They also seem to show different effects, and we've also looked at other types. We've characterized the mechanism, as you can see on the right, and we have really pinned it down to lysosomal signaling that then inhibits apoptosis and apoptotic pathways. And just to put this in perspective and its therapeutic importance, why do we care? This is really a simple and easy way to enhance lung-based or macrophage-based therapeutics. We're a pulmonary drug delivery group, so we're using this for lung cancer, where we're transplanting macrophages that carry particles and have anti-cancer benefits. And with that, I'd like to thank you for your attention and thank INBRE and the University of Delaware and Pharma Foundation for funding. Thank you.
Excellent. Thanks very much, Peter. That was a great talk and right in my wheelhouse, as we also do similar research. So next up, I'm pleased to introduce Mitch Gravely from the University of Rhode Island. And Mitch, you can go ahead when you're ready.
Hi, so my name is Mitch. I'm a PhD student from the University of Rhode Island, and today I'm going to be talking about multispectral fingerprinting to resolve nanomaterial trafficking in endothelial cells. And so the nanobio interface is the main site where a lot of interactions occur that dictate how cells interact with, internalize particles, and where they end up and how they're processed, and they signal different pathways within the cells. And so are a couple of components that are very important to this, and it's the suspending medium, the nanomaterial itself, there's a solid liquid interface, and then of course the biological substrate. And it's where all of these sites meet that the nanobio interface occurs. And so, as I just mentioned, these interactions ultimately dictate whether a material is biocompatible or can promote a bioadverse outcome. And so we must fully understand these interactions if we ever want to be able to predict and prevent nanotoxicity for new developed materials. Next slide, please. And so in our lab, we use DNA-wrapped single-walled carbon nanotubes, and these are biocompatible and readily internalized by cells by active endocytosis. And so these translocate into endosomes and lysosomes of mammalian cells, making them just naturally locate to a spot where we can look at intracellular trafficking. And so these exhibit exceptional optical properties, including NIR fluorescence, which is sensitive to the local environment. So different solution properties or analyte binding produce different spectral responses. And at the same time, resonance-Raman scattering, which can tell us about concentration, aggregation of particles, and surface chemistry. And so in this approach, we combine these optical spectroscopy methods into a microscopic approach to look at single organelles of cells and be able to comprehensively investigate intracellular trafficking processes that are occurring. So next slide, please. And so to summarize a lot of work into one single slide, we were able to find that nanotubes, they respond to endosomal maturation and the processes that occur during endosomal maturation. For example, there's a shift in fluorescent spectra as the pH decreases and then decreases further as nanotubes move into lysosomes. And at the same time, aggregation is constantly occurring of particles that have been internalized by these cells. And so we were able to observe all of these and come up with a sensor for endosomal maturation. And this could have a significant impact on future studies where we can look at different particle wrappings, particle types, and be able to tell how these affect these maturation processes that are native to the cells. And so with that, I'd like to thank my funding sources, my lab mates, and everyone who's helped with this project. Thank you.
Excellent. Thank you, Mitch. So for those of you listening, we are welcoming questions. So if you want to type in a question in the ask a question on the screen, that would be great. But in the meantime, I'm pleased to introduce Daniel Keene, who is also from the University of Rhode Island. And Daniel, it is time for you to begin when you are ready.
All right, I'm ready. Hello everyone. My name is Daniel Keene, and I'm a PhD student at the University of Rhode Island, working with Dr. Ryan Pulling-Skovic. I'll be presenting on physically cross-linked emulsions to mimic the structure and mechanics of biological tissue. And as I'm sure all of you know, biological tissue can be very challenging to work with due to the challenges of precision studies, due to all of the various interacting components of cells and tissues alike, in addition to the challenges of the transport and storage of said tissue. Therefore, it's of great interest to produce a synthetic mimic to this biological tissue. Next slide, please. So the main question I'm trying to answer is, can the compartmentalized structure and elastic intercellular connections of biological tissue be synthetically replicated? So biological tissue can be simplified to just individual cells connected by the proteins of the extracellular matrix. I'm attempting to create a synthetic system in which oil and water emulsions replicate the cells, and triblock polymers create the intercellular connections that would ordinarily be produced by the proteins. The polymer that I'm working with is polystyrene ethylene oxide styrene, or CEOS, which is an amphiphilic polymer in which the end blocks are hydrophobic and the middle block is hydrophilic. This results in the polymer taking one of two conformations, either loops, in which case both end blocks go into the same dispersed droplet, or it forms bridges in which each block goes to a different droplet, connecting these two elastically. And the theory that bridging is primarily occurring can be supported by both rheology and dynamic light scattering. In rheology, you can see that as the concentration of CEOS increases, both the viscosity and the elastic behavior of the material increases, especially compared to ordinary PEO in the system. The systems containing CEOS are far more elastic. Furthermore, in dynamic light scattering, a clear bimodal distribution is seen, in which individual droplets and clusters are observed. Next slide, please. So to conclude, there's ample evidence that bridging is occurring, and the compartmentalized structure and elasticity can potentially be replicated. The moduli of brain tissue is approximately 1 kilopascal, and we're reaching that moduli easily. As I move forward, I'd like to study the transport of particles through the medium, examine temperature-dependent behavior, and examine neutron scattering to explore dynamics of polymer chains. And thank you for listening.
Excellent. Thanks, Danny. All right, next we are transitioning to hear from Kalindu Pereira from the University of Rhode Island. And Kalindu, you can go ahead and go when you are ready.
Yep. Oh, thank you. Okay, well, good afternoon, everyone. My name is Kalindu Pereira. I am part of the Menon Lab here at URI. And so today I'll be taking you through some tissue engineering work that we did and published and that we're continuing to do, namely the PiniPalm microwell arrays. So there's been this kind of slow shift in the past 5 to 10 years away from these not really physiologically relevant cell cultures in these flat 2-dimensional well plates to more 3-dimensional cell cultures, especially for cancer drug discovery, where you need this solid tumor mass to really get a good idea of whether your drugs work or not. You can see those spheroids at the bottom of the screen, along with an illustration. And there's a number of ways of doing of making these spheroids. I've shown you one, which is the hydrogel at the corner of the screen there. It's made out of agarose. It's got these little microwells, and it's perpetually hydrophobic, which means the cells can't really attach to anything other than themselves, forming the spheroid. Next slide, please. So our goal was to kind of build on this technology and get a hydrogel with microwells that could form spheroids of uniform size, which are also easy to harvest once we have the spheroids made. So what we did was we used a thermoresponsive polymer called PiniPalm, which has a tunable property, the LCST, a temperature above which our gel and our wells shrink and become hydrophobic, essentially allowing the spheroids to form and compacting them. And below that temperature, the gel and the wells expand, allowing easy harvesting of the spheroids that we've made. In the top right-hand quadrant, you will see how we made these gels with a hard polymer mold. And at the bottom left, you will see that we got the LCST exactly to about 35 degrees Celsius. And you can see how the gels and the wells look below and above that temperature, the different sizes of the wells in particular. So we looked at a number of different cell lines, cancer cell lines on these wells, perfectly cytocompatible, good uniform size for the most part. Next slide, please. But there are limitations. One of the main ones is that it takes a long time for us to pop these gels out of the mold, high failure rates. So now what we're doing, we're having a surf undergraduate work on this, is to come up with a photo ink that'll be compatible with this 3D bioprinter that we've got, and just to print the gels, as you see on the screen, in situ without the need for a gel. And now we're moving on to try and mimic the physical and mechanical characteristics of individual tumor tissue, ECM. And so stay tuned. More of our research can be found at the link at the bottom of the screen. Thank you for listening.
Excellent. Thank you very much, Kalindu. That was great to hear. So next up, we have Matt Freeman from the University of Rhode Island. And Matt, you can begin when you're ready.
Thank you. Good afternoon, everyone. I'm Matt Freeman from the University of Rhode Island, and today I will be giving a brief presentation on a project titled Rifampicin Cyclodextrin Inclusion Complexes Formulated for Pulmonary Drug Delivery, specifically for the treatment of tuberculosis. Tuberculosis is still extremely prevalent today, with 10 million new cases reported annually and more than 1.5 million tuberculosis-related deaths. One of the main issues is the current treatment plan involves an oral dosage form that is taken frequently and for a prolonged period of time. The length of treatment, which is usually 6 to 9 months, leads to poor patient compliance, while the oral dosage form results in systemic delivery and potential hepatotoxicity. So in this project, we were seeking to engineer a microparticle capable of direct delivery to the alveolar region of the lung via a dry powder inhaler, allowing for improved treatment efficacy of tuberculosis. Next slide, please. To accomplish this, we investigated spray-dried rifampicin cyclodextrin inclusion complexes. By doing so, we can increase rifampicin solubility, and by doing so, we can increase the amount of rifampicin we can deliver to the lungs via complexation, while also producing particles capable of deep lung delivery. While not all the data is shown here, we confirmed rifampicin cyclodextrin complexes remained intact after spray drying, and the formulations displayed the appropriate aerodynamic diameter for deep lung deposition. The next step was we wanted to incorporate a nanoparticle that specifically targeted alveolar macrophages. And by doing this, we created what we call in our lab nanocomposite microparticles. So the rifampicin cyclodextrin complexes made up the bulk of the particle, while the nanoparticles were within the complex, or within the rifampicin cyclodextrin complex bulk material. And the nanocomposite microparticle formulations allowed for deep lung delivery of rifampicin cyclodextrin complexes, in addition to rifampicin-loaded nanoparticles that provide us a sustained release and pH responsiveness. Next slide, please. The previously described nanocomposite microparticle allowed for the delivery of nanoparticles to the alveolar region of the lung, which takes advantage of nanoparticle penetration and macrophage uptake, while the bulk rifampicin cyclodextrin complexes provided a rapid release of rifampicin. Spray drying was used to develop the nanocomposite microparticles to target delivery to the lungs. This potentially reduces the required dose and reduces off-target side effects. Furthermore, spray drying is an easily scalable process that provides control over various particle characteristics. Finally, the inhalable dry powder provides patient ease of use compared to alternative lung delivery methods, such as a nebulizer. This, combined with the direct delivery at the site of infection, has the potential to dramatically improve treatment efficacy of tuberculosis and overall patient outcomes. Thank you, and please feel free to email me if you have any questions. And I just want to thank all my lab mates, my advisor, Dr. Munich and Dr. Shen, and our funding sources.
Excellent. Thanks, Matt. We are still accepting questions for those of you in the audience, and including our presenters. And next up, we have Tamar Abel from Dartmouth College. And Tamar, we're excited to hear your presentation when you're ready.
Hi, I'm Tamar Abel, and today I will be presenting on the single-cell chromatin heterogeneity of fibroblasts from a systemic sclerosis 3D skin-like tissue model. I study a disease called systemic sclerosis, or SSC, which is a severe multi-organ autoimmune disease and unfortunately has very high mortality and currently very limited treatments. In the clinic, it presents with both vascular and immune dysregulation, as well as extensive fibrosis. Some contributing factors are thought to be genetic predisposition, as well as an environmental trigger that causes onset of the disease. However, lack of a strong genetic association suggests that molecular mechanisms which act at an epigenetic or non-coding level are likely the most influential driving factors for this disease. A method commonly used for studying epigenetics is ATAC-seq, which is able to probe chromatin accessibility by the insertion of a TN5 transposase into open regions of chromatin. These can then be sequenced and mapped across the human genome. We use single-cell ATAC-seq so that we can look at chromatin heterogeneity within cell types such as fibroblasts. Our goal is then to use single-cell ATAC-seq to determine the chromatin patterns and epigenetic factors that are enriched or depleted in SSC fibroblasts, which are the key effector cell type for this disease. We hope the benefit will be novel molecular targets for more effective treatments for this disease. Next slide. We apply this technique to our 3D tissue culture, which is a novel disease-specific model for SSC. And we do this by incorporating many components directly from patients. So we have fibroblasts, macrophages, keratinocytes, and also patient plasma in our 3D tissues. Over the course of 5 weeks, we are able to effectively grow human skin in vitro. We then are able to see from H&E staining our fully formed tissues, as you can see on the left-hand side. And you can see that these have 2 layers, as we would see in human histology from skin. And we can also see that the SSC tissues have a slightly more contractile phenotype. If you look at the right-hand side, here's our single-cell ATAC-seq data from these tissues projected onto UMAP projections. And 2 key things from this, so on the top 2 panels, we were able to determine cell type using key transcription factors for known cell types, as well as to find 5 distinct groups of fibroblasts in the bottom 2 panels as predicted by predicted transcription factor activity. Next slide. Performing fold enrichment on these clusters helped us to find that these particular clusters of fibroblasts, as characterized by unique epigenetic states, are enriched or depleted in an SSC disease-specific manner. So in conclusion, we were able to identify 5 epigenetically unique groups of fibroblasts from these 3D tissue models. We were also able to find that these are enriched or depleted in an SSC-specific manner. And next steps will include expanding our data set as well as finding targets for further studies. I'd like to thank the patients with SSC and their families, as well as the Center for Quantitative Biology and the Whitfield Lab at Dartmouth, as well as other funding sources. Thank you.
Excellent, excellent talk, Tamar. Next, we're going to be moving on to hear a talk from Sabrina Delva, who's also from the University of Rhode Island. Sabrina, we're ready for you to begin when you are ready.
Can everybody hear me?
Yes, you're good to go.
Okay. Hi, everyone. My name is Sabrina Delva. I'm from the University of Rhode Island, majoring in biomedical engineering. So today I would like to talk to you guys about the evaluation of nebulized lipid-coated nanoparticles for sustained pulmonary delivery of chemotherapeutics. So the purpose of this project was to develop and evaluate nebulized inhalable core shell nanoparticle for the formation in terms of its physiochemical properties as well as its in vitro efficacy. And we did this using a commercially available nebulizer called AeroNeb. Next slide, please. So nanoparticles have been becoming more popular for treating site-specific and long-term therapy for chronic illnesses. Biodegradable nanoparticles are excellent for this because you can offer a sustained drug release at a pace that corresponds to the polymer breakdown, as well as their ability to degrade inside the body without leaving harmful byproducts. Nebulizing the nanoparticles would allow drug-loaded nanoparticles to enter the body directly towards the lungs. So at the top, we have the comparison of the DLS measurements, both pre and post nebulization. And as you could see, the particle diameter, as well as the PDI and zeta potential, have shown no significant change after nebulizing the nanoparticles. And to the right, we have, oh wait, I'm hearing an echo. Can everybody hear me still? Okay, I heard an echo, sorry. Oh wait, can you bring it back to the other side? And to the right, we have a picture of the SEM images of the nanoparticles, and you can see that their morphology has been preserved through nebulization. And at the bottom, we have an FTIR of the nanoparticles, which is confirming the chemical composition of the nanoparticles was maintained after passing through the nebulizer because there's no change in the peaks. Next slide. So, sorry. The drug release at the top shows that after about 50 hours that the paclitaxel was released from the nanoparticles, and it was released at a sustained pace for the rest of the duration, and 84% of the drug was encapsulated by the PLGA nanoparticles. And we performed a cytocompatibility study using the E549 cell line to determine the biocompatibility of the nanoparticles. And you can see that they are compatible in nature. And we did this using an MTT assay as well. So the next steps of this project would be to perform in vitro studies using rats, as well as using other drugs to support more in vivo studies with the nanoparticles.
Excellent. Thanks, Sabrina. Next up, we have Yahira Rivera Lopez from Delaware State University. And Yahira, we are ready to hear you talk when you are ready.
Good afternoon. My name is Yahira Rivera, and the title of my research is called Raman Spectra of Concentrated Glycol Solutions. In earlier times, scientists overlooked the importance of observing molecules in a crowded environment. Now we see these crowded molecules in our day-to-day lives, such as in separating blood cells or even in our islands. But what is macromolecular crowding? Macromolecular crowding can alter properties of certain molecules when there's a presence of high concentrations of macromolecules like proteins, lipids, or carbohydrates. Along with this, macromolecular crowding can strengthen the bond between protein and DNA. It assists in transcription, DNA replication, protein refolding, or binding of small molecules. In this presentation, I spent my time learning about the Raman spectrometer and how to apply it towards FICO, which is a neutral, highly branched, high-mass, water-soluble polysaccharide crowding agent. I used the Ariba Raman spectrometer to measure the Raman spectra of the quartz cuvette with H2O, and quartz cuvette with different concentrations of FICO ranging from 100 mg per mil to 1,000 mg per mil. Next slide. Figure 1 displays the Raman spectra of the cuvette from H2O and the FICO concentrations. However, we're going to focus on figure 2 because it displays the same spectra of the previous figure, but the range has been zoomed to 1500 to 3500 wave number to avoid spectral contamination from the cuvette. Figure 2 also allows us to see the dissolved peaks from the nitrogen and oxygen, the peaks associated to H2O, and lastly, we can see some peaks that can be associated to FICO. We hope that we can use these peaks as signatures to identify FICO against other materials. Next slide. We're excited to say that the peak at 2900 wave number can be associated to FICO because, as we can see in figure 3, the graph displays an increased intensity of the 2900 peak when the concentration of FICO enhances, unlike the surrounding molecules from H2O or the quartz cuvette that are displayed in figure 4 that without a doubt began to show water restriction, and it resulted in the decreasing of their peaks. Nevertheless, for future work, we hope to complete more analysis on diverse spectral signatures and apply it to the studies that observe crowded environments. Thank you.
Thank you very much, Yara. Next up, we have Celestia Tulador from Keene State College. And you can begin your presentation when you're ready. We're excited to hear what you have to say.
Sure. Can you hear me?
Yeah.
Okay. Hello, everyone. I'm Stacia Leather, and I'm an undergraduate student studying at Keene State College. And today I will be presenting you on the rheological investigation of pre-cross-linked hydrogels for 3D bioprinting process. So what is 3D bioprinting? It is a revolutionary technology that uses a computer-controlled 3D printing discipline to release biomaterials layer upon layer along with high-precision positioning of cells. The way it works is groups of cells are taken from host body, mixed with hydrogel, and this is what we call the bioink. Using bioprinting technologies, they are printed, cultured, and therefore tissues are regenerated. As thousands of people wait for organ transplantation every year, this technology has a huge application in regenerative medicine. But why 3D printing with extrusion-based 3D printer? The bioink is subjected to certain pressure, which challenges the viscosity of the hydrogels in several phases. At rest, during the extrusion, and mainly the retention phase of the printing. So we use rheology, which is studying the properties of liquid to predict how it works in the 3D printer to narrow down our sample space. Next slide, please. So in this research particularly, we focus on the retention part of the 3D printing, ensuring the proper 3D shape fidelity of the scaffolds by cross-linking them. We can either pre-cross-link the scaffolds or post-cross-link them to change them from gel to more firm consistency. Some of our past researches and findings from post-cross-linking suggested poor shape fidelity and motivated us to try pre-cross-linking the hydrogels with calcium chloride. For the material preparation, alginate and carboxymethylcellulose were mixed at 8% of solid content, 1% and 0.5% of calcium chloride was used, and sets of rheological investigations including the flow curve, 3I TT, and amplitude tests were done. The results from the flow curve shows exactly what we wanted to see. Upon the application of pressure, the viscosity decreases even while pre-cross-linking. Next slide, please. The 3ITT shows the recovery rate of each compositions with time. For example, for 0.5% of calcium chloride, A8C0 recovered about 100% in 1/22nd. We went further into printing them by setting the given parameters on screen. As a conclusion, the scaffolds were cross-linked all the way through and likely to show better 3D shape fidelity. Thank you so much for joining me in this presentation, and I would like to thank Keene State College, New Hampshire Biomate, New Hampshire INBRE, and IDEA for funding and supplying us with equipment, and I am up for any questions that you have about this presentation. Thank you.
Excellent. Thank you very much, Masha. That was a great talk. Last but not least, we are going to bring in Bendeshwar Saw from the University of Rhode Island. And Bendeshwar, you can go ahead and begin when you are ready.
Yeah. Hi. Okay. Okay, we have a title like a effect of the size of the nanoparticle size, radiation energy, and targeted nanoparticle for you. So copper cismin nanoparticle, a newly invented particle. It is not available market, but it was a very effective for the radiation therapy for the most of the cancer cell. So our purpose for that experiment, like which size and which energy level get produced like a higher singlet oxygen. That means if you get a higher singlet oxygen, that means they kill the more cancer cell. So that is a purpose. So we have used like a 14 nanometer, 100 nanometer, and 200 nanometer with the copper cismin nanoparticle and radiation energy like 90, 250, and 350. And also we have using the same nanoparticle with the targeted, means target hyaluronic acid. That means if you have conjugated with the nanoparticle, they can move faster than without like a non-targeted nanoparticle. So that is the purpose we have to use the hyaluronic acid like a targeted molecule in the nanoparticle. And then obviously we have to use the different radiation dose. So that means dose means like a control means like a no radiation, just we have to put, but 8 means 8 gray radiation for the X-ray. Next slide, please. So here we have a follow in the like a singlet oxygen. We have measured like a 2 different batch of the nanoparticle. And we found that we have in the left one, we have like a 90 kilovolt and absorbance measurement. Similarly, we have to measure the 250 kilovolt, and the third one we have like a 350. So we found that overall for the three of them, like a lower energy level produced like a more singlet oxygen compared with the higher energy level. So one of the factor could be they have like a 200 produced more like a 200 nanometer of the radiation with the radiation therapy produced more singlet oxygen than the 40 and 100 nanometer for the first batch. So even we can compare with the obviously in the 90 kilometer 90 kilovolt has a targeted nanoparticle higher, but compare with the 250 and 350 overall, I would say the same as like a higher concentrate, higher per size of the particle get a more produced. So the reason for that means they have like a more like a higher particle size. So next one, please.
We have about 15 seconds.
Oh, so last one, we have like a second batch of the experiment, and we have done again the same process with the same methodology. So again, we found the overall, we have like a 90 kilovolt get a higher significant than like a 250 and like a 350 nanometer. And obviously size was not too much a difference, like a significance difference for that one. So overall, we would say like a 40 and 200 nanometer size get not too much difference, but a significance compared with the 100 nanometer. Thank you.
Thank you very much, Bindeshwar. So now I don't think we have our final speaker here, likely because of technical difficulties. So we are going to move on to a couple of questions that I have for some of our presenters before wrapping up this session. So this first question is actually for Mitch Gravely. And the question is, was a general not wanting to know, what are the next steps in your research?
Thank you. That is a very good question. And so we've spent a lot of time developing this method to really be able to see inside what's happening within a cell. And so part of the next steps of our research is to continue on that path and translate some of these findings and some of these things that we can see to nanotoxicity and potential changes that are occurring. Because that's the main goal, is to relate, if you have one type of particle wrapped with one type of, surface chemistry, for example, does that alter the toxicity of the compound? And so we know on some level it does, but generally we want to see that within the cell and in real time. Excellent. So I hope that answers your question.
No, I think that was a great answer, Mitch. And then the next question is for Bader Jirai. And the question is, what is it about the PEG coating of your nanoparticles that enhance survival of macrophages, and what would other nanoparticle coatings do in this case?
Absolutely, that's a great question. And what we have, actually our nanoparticles are not coated. They are made consistent, the whole nanoparticle is PEG based. And so we have shown that, we've used that as our base particle because some of the data that I've shown that they don't cause any pro or anti-inflammatory changes to the macrophages, because PEG has been characterized as bioinert in the literature. And so that's why we chose that in the beginning. But we have also investigated a wide range of nanoparticles: silica, gold, silver, polystyrene, and we're considering others just to show that all of these nanoparticle chemistries are able to enhance survival. So it's really not about the type of nanoparticle itself, but the act of internalization that then triggers these pro-survival signaling and suppresses apoptosis that we see. So it's going to be a differential based on the chemistry and some of the other nanoparticle properties, but in general, all of the nanoparticles were able to show that. Great question.
Excellent, thanks, Bader. So everyone, that is all the questions I have for the technology session. To all of the presenters, you did really excellent work and gave really excellent presentations. And I thank you for your time and your enthusiasm and your patience in the submission process and getting your presentations ready together today. So now we are going to take a break until our next session, which will be our psychology and health lightning talk session. Thank you.